Well, good afternoon and welcome to Life Science Washington's DeviceConnect series, Novel Delivery Mechanisms for Modern Medicine. We are pleased to present you this series in partnership with Product Creation Studio. I am Dale Willard, Director of Member Services for Life Science Washington. For those who are unfamiliar with Life Science Washington, we connect our over 500 member organizations to each other and to funding, talent, partners, and essential business services. We elevate our industry by promoting policies that strengthen the environment for groundbreaking research and product development that improves and saves lives. I'd like to extend a special thanks to our sponsor and co-producer of this event, Product Creation Studio. This event is free because of their support and commitment to our industry. I also want to encourage everyone to attend this event's virtual networking immediately following the panel discussion. It will be your chance to meet tonight's panelists plus fellow audience members virtually. I'll give more directions at the end. First, a couple of housekeeping notes. One, for those in the audience, please double check that you're muted and you can stay muted. And number two, the goal will be to take a few audience questions at the end. And if you have any questions, please submit those via the chat box. All right, so allow me to introduce today's cast of speakers. It is an honor to have with us today Dr. Saniya Ali, Chief Technology Officer and co-founder at Watershed Therapeutics. Also, Professor Deborah Fuller in the Department of Microbiology at the University of Washington School of Medicine. We also have Manjari Lal, Dr. Manjari Lal, who is Program Advisor at PATH. Last, but most importantly, I want to introduce you to our moderator, Scott Thielman, Chief Technology Officer at Product Creation Studio. Scott and PCS, our Product Creation Studio, have long been partners in the creation and organization of the DeviceConnect series and a valued partner. Scott is a co-founder of Product Creation Studio, a medical device design and prototyping firm, and an active resource in our medical device community. So Scott, are you there and ready to take it away?
I am. Thanks, Dale. And thanks to Life Science Washington in general. Hopefully you guys can hear me. And we've got folks like Dale, Craig Matthews behind the scenes, and Amanda over there that we're working with and are working with us to put these presentations on, these panels. So much appreciated. And this was in amongst their busiest time of year with the Life Science Innovation Northwest just happening a couple of weeks ago. So thank you guys for all the attention and ability to get this event out. So for a brief history, we've been doing these events that we call DeviceConnect for, boy, I think we were pre-pandemic for sure. So maybe going on 3 or 4 years. And the focus is to get folks from kind of the various reaches of the medical device space, life sciences space, out and talking to one another on various topics that may be relevant to them. And we always do that with a nice cross section of folks on a particular topic. And so we're excited to have this panel today. But my company and our reason for interest in this particular topic is that we've worked in the drug delivery space and clinical workflows for quite a while at PCS, and we've seen the complexities that can be in there, the subtle complexities sometimes, either with the user or with the technology itself that must be delivered either in a targeted way or systemically in a way that does the magic that we all hope is going to happen. And so we wanted to dig a little bit deeper into where our healthcare is heading here with some of these innovations. We've seen innovations in vaccine recently, notably with the COVID vaccine, how a couple companies were able to turn on a dime basically and get a vaccine candidate out very rapidly with some of the novel nucleic acid vaccine technology. And then we also see other examples of compounds of interest getting to patients that need them. For instance, chronic diseases being managed by continuous glucose monitoring, insulin pumps. We're seeing cancers being treated with targeted radiation, targeted immunotherapies, gene therapies. All of these things have challenges on the technical side as well as the user and user interface and design side. And so very excited about the potential, but also the challenges that have to be overcome as we move forward. And I think one thing we can say for sure that we're lucky in this country to be able to say is that I think we can be legitimately hopeful as patients in the modern era, with a tip of the hat towards, hey, we realize we just lost about 1 million people to COVID in the last 2 years, but on the other hand, it could have been much worse, and we were able to mitigate some of that, and so I believe we can look at our healthcare system now and often find that there is a potential solution, or there's something that might mitigate our disease and help us along. So we're going to hear a little bit more about from several of the innovators on the forefront of delivering modern day medicine. And we already heard a brief description. So we'll have Saniya Ali from Watershed Therapeutics. Now, Saniya's got a biomedical degree from Duke, but also went to Stanford's Biodesign program for postdoc. So she's going to kind of be our user-centered designer turned drug developer today, and we'll get to learn a little more about her company that's solving large unmet needs in the challenges around urology. And then Deb Fuller from the University of Washington, Department of Microbiology. And Deb's got so many titles that I can't list them all, but she's also Associate Director of Research at the Washington National Primate Research Center. And has been a driving force behind developing nucleic acid-based vaccines and antivirals that include DNA and RNA vaccines for infectious disease. She's led at least 2 of these from the bench to clinical testing, and including one of the only DNA vaccines to induce protective levels demonstrably in humans. So Deb's also our pundit. You may have seen her on Bloomberg TV, CNN, NPR, Washington Post, et cetera, et cetera. She's been tapped quite a bit for her kind of plain speech on how vaccines work and what we should be looking for there. And then Manjari, Dr. Manjari Lal is a program advisor at PATH, and she's been focused on formulations for pharmaceuticals, for vaccines, primarily with a focus around low and medium income countries. So how do we bring these technologies, how do we formulate them to be feasible for the case of world health? And so Dr. Lal is going to be our global health formulations expert with a wide variety of experience on trying to solve the hard problems that come about when you try to disseminate some of the technologies out to the world where we're away from some of the resources that we normally have in the industrialized world. So let's dig in and start hearing from our panelists. I want first question, want to just kind of get a little bit at where our panelists come from and kind of what drives them to be in the fields that they're in. So, but Manjari, let's start with you. And I believe we used the qualifier modern in the description of this title of the topic, and it's kind of my own bias here comes from living in the United States. But if as you focus on delivering solutions well beyond our borders, what does delivery of modern medicine mean to you and to PATH as you guys look at the global health needs?
Thank you, Scott. So just for the audience, before I take a deeper dive into that question, if you're familiar with PATH, we are a global health nonprofit headquartered in Seattle. And we have offices, I believe, in 30 or 40 countries worldwide. And for more than 40 years, PATH has been working with communities around the world to improve and accelerate access to health innovations. At PATH, I lead the formulation portfolio with focus on formulating vaccines and pharmaceuticals to improve access, acceptability for in low and medium income countries. And in formulating these vaccines to make them more accessible, more acceptable, we look at a multitude of technologies. And the goal here is that, one, to identify technologies which are suitable for a specific vaccine candidate, but also the technology should be easily adaptable, adopted, scaled up by the manufacturers who are in the developing countries or low and medium income countries. So that is something which is really wired into this our process, our formulation process. Because I think, to answer your question, Scott, there are the innovations usually happen in rich nations. I would say the innovations happen there, and for countries outside of US and EU, it is difficult for them to gain access and enjoy the benefits of these innovations, because the cost is a huge hindrance. And also the scalability, the manufacturing process, all those things are, I would say, a hindrance in adoption. So PATH is constantly striving to find ways, alternate approaches to bring the cost down, to make the technology more accessible. One very good example is the RNA vaccines from Moderna and Pfizer. In US and EU, refrigeration is not a challenge. It is not difficult to freeze the vaccines using ultra cold freezers, or I would say maintaining a cold chain throughout is not a big deal. However, it is a big deal in low and medium income countries where there are constant power fluctuations, continuous refrigeration cannot be maintained. And that is where we come in. We try to fill that gap by addressing some of the things which are low-hanging fruit. Can we make the vaccine more thermotolerant? Can we make it more thermostable so that it can be distributed? Equitable access, I would call it, making it available to everyone in the world. So I think that is where we come in. That has been working in this area for quite some time, making sure that vaccines are equitable, easily accessible, and everyone can lead a healthy life.
Right, exactly. But making them thermostable, if that were easily achievable, would be something we would all welcome, right? So obviously at PATH, you're taking the hard road and trying to solve some big challenges, right? Not just deliver the same formulations as we have here.
Right, and I also want to add that, I mean, thermostability or improvements in thermostability is one aspect we look at when we are conducting a technology assessment or trying to find a technology which is well suited for a specific candidate. Thermostability is one aspect. And there are other factors also, for example, the regulatory pathway, the scalability of the technology. the COGS. I mean, there are all these factors which go into consideration when we select a technology. Specifically from formulation point of view, yes, thermostability is one of the criteria we look at. We also look at how the devices, delivery devices, are they compatible with the drug or vaccine candidate which it is holding in? Is there any leachables or extractables we should be concerned about when we are housing the vaccine antigen in the device? So those are all formulation considerations, I would say, we look at when we are evaluating a delivery device.
Got it. Okay, since we're talking vaccine, maybe we'll shift over to Deb. Deb, we've known each other for some years now, and I think the entire time you've spoken and talked about nucleic acid vaccines, some of the modern vaccines, and really I felt like it wasn't until the pandemic that a light bulb went off in my head. It's like, oh, this is what Deb's been talking about, and so for many of us, an mRNA or a DNA vaccine is something totally new and somewhat magical. But you've been dealing with these or working towards these for a large part of your career. Can you kind of put these recent advancements in context for us? What are we looking at when we hear about Moderna and Pfizer producing a vaccine within a year, say, and getting it on market?
Yeah, I always like to tell everybody that the COVID-19 and the development of nucleic acid vaccines was like a perfect storm, that they both hit each other at just the right timing. Nucleic acid vaccines have been studied and were really discovered to be feasible over 30 years ago, back in 1990. That was when I actually got started with the technology, where we actually figured out a way to directly inject DNA into plant cells. And then, light bulb went off in our head, thinking that maybe we could do this into animals and in mammalian cells. Initially thinking it was gene therapy, right? And I think a lot of people thought about using RNA and DNA vectors and injecting them into in vivo from the perspective of a gene therapy approach. But it wasn't really very effective for gene therapy, because if you think about what gene therapy needs, it really needs most of the cells of a given tissue to express that particular gene. That just isn't how DNA and RNA vaccines work. You can't, right now there's no technologies that really deliver them that way. But what we discovered along the way was this pesky problem. We kept getting an immune response against the thing we were expressing from the DNA and RNA. And then another light bulb went off, and we realized that, golly, this is an entirely new class of vaccines. And it really kind of took off from there, or in the early 1990s. By 1998, the first phase 1 human clinical trial was already starting with a DNA vaccine at the time. And so it was an exciting time, because we could foresee really a future. If you think about what DNA and RNA vaccines are, they're simply, all you need when you have a new outbreak or a pandemic or any sort of infectious disease is the genetic sequence of that particular pathogen. And rather than having to say produce a vaccine in a lab and go through this long process of purification and the expense and the like, instead you turn a body into its own factory for producing that vaccine by injecting that nucleic acid into the cells. And so the concept was just so exciting. Everybody at the time, back in the early 90s, foresaw a future where DNA or RNA vaccines could replace virtually every vaccine out there, but also solve the problem of generating vaccines for infectious diseases that we didn't have vaccines for yet. And so that was the vision, and that was 30 years ago. But we ran into a lot of problems. And the first and foremost was that the RNA vaccines were too reactogenic and too unstable. And the DNA vaccines were too poorly immunogenic. And central to both of these, the biggest problem was delivery, because the one thing that you got to do with DNA and RNA vaccines is you've got to get them inside the cell. And how do you do that? And so this is where devices became, essential, and that's sort of where it led into the area of research that I'm working on now, and how better to deliver DNA and RNA vaccines efficiently into cells. But 30 years later, fast forward, and all of a sudden things like lipid nanoparticles really only started to gain traction in the last 5 years. And all of a sudden RNA vaccines started taking off because you could formulate RNA vaccines with lipid nanoparticles. And so just when COVID-19 hit, there were a number of groups, obviously Pfizer, Moderna among them, and BioNTech with Pfizer, who had gotten to the point where they were actually starting phase 1 human trials with for other infectious diseases like influenza, like RSV. They had solved the problem of delivery and stability. They were seeing great immunogenicity in animals, and they were moving into phase 1 and even 2 human clinical trials for other infectious diseases. And then suddenly COVID hit. And all of a sudden, all of those thinking back 30 years ago, we thought, well, this would be a rapid way to respond to emerging pandemic because all you need is a genetic sequence and you go, boom, quickly make a vaccine. We were put to the test. These vaccines were put to the test, and they actually, they met the challenge, to produce and license a vaccine in less than a year is just, that is revolutionary. Okay, and so now, the way I see it going forward is that DNA and RNA vaccines, they're not just, there's just a whole new revolution ahead of us in terms of things we can do. We can talk about that later, what else we could use them for. But that's kind of where I've seen it,
right.
And it's just exciting to have been part of this area of research for all this time and watch this evolution right up to this this exciting moment, and yet we've got more to do.
Yeah, well, we're glad that you and others are working in this field all of a sudden because of the potential benefits here that we've had. So the just to clarify, so even with the LNP delivery mechanism, it's still really all the game is about getting that nucleic acid, the DNA or the RNA into the cell, and after that, the cell kind of takes over and expresses it does the rest.
Exactly. Yeah. Once the RNA or it gets inside the cell, then it is read by the cell and it actually produces a protein. The RNA doesn't stick around; it disintegrates, but the protein is now produced, and so your own cells become the factory producing your own vaccine. There's a lot of really important benefits to that, such as for example, when you have to produce a protein or a recombinant protein in a lab for a vaccine, you have to make sure that it's conformationally okay, that it's purified. And a lot of times there can be a lot of problems. That protein can fall apart in the lab. But if you actually have your own cells producing, it's right in vivo, and you have that immediate production of that protein in its natural conformation, with presented immediately to your antigen presenting cells. And there's just a lot of different types of things that we discovered. For example, that RNA and DNA vaccines are capable of not only triggering the antibody response, which plays the major role in protection for vaccines, but also triggering a backup immune response called a T cell response. And if the virus suddenly mutates and starts to evade our antibody and gets inside our cells, these T cells can then find those infected cells and eliminate them before they can cause disease. And we think that to some extent, the fact that these types of vaccines can leverage both types of immunity is one reason why a lot of these, even though we haven't updated the vaccine for COVID-19, that is still sustaining excellent protection against severe disease in the population. So, but there are limits, and Manjari brought up one important one, is that those lipid nanoparticle formulated RNA vaccines are not very stable when they're at warmer temperatures. I don't know about how many people got a call and said, "Hey, you want a vaccine? This thing is thawed out. We'll have to throw it away if we don't get enough people coming over to take it." That's a big issue, especially for distribution. And there's just, as Manjari pointed out, there's just areas of the world where the vaccination rate is very low simply because of the requirement for this cold chain and the difficulty in being able to distribute vaccines there. And the lipid nanoparticles are one of the formulate RNA vaccines have very high bar because they require that deep freeze. And so that's a big issue that is now a lot of scientists, including our own group, are working on to try to solve.
Right. Okay. Well, it's good to hear the reasons why all that was going on. I clearly remember the need for deep freeze was a major consideration. Well, let's and we'll get back to, I think shortly we want to hear about alternate ways of delivering some of these vaccines, but I want to shift over to Saniya. Saniya, you're a medical device expert, biomedically trained, and so I think of it as a device person for the most part, but you're actually leading the development at a drug company. And so maybe tell us a little bit about the story behind Watershed and what you're up to and how you got from kind of a Stanford biomedical focus to being a drug company.
Sure, absolutely. So like every good company, our company, Watershed Therapeutics, really started from an observation that uncovered a major discovery of a huge problem that exists currently in medicine. My colleague and I, my co-founder and I, we are both alums of the Stanford Biodesign Innovation Fellowship, where we had the opportunity to really immerse ourselves in the entire medical care system at Stanford. And the whole purpose behind that is to observe surgeries, observe procedures, observe patients, at their lowest moments, observe physicians treating these patients and identify huge unmet clinical needs. And one of the observations that kept coming up was in an ER setting where we saw a postmenopausal woman come in with symptoms of stroke. She was experiencing fatigue, dizziness, could not really communicate or talk. And after hours of workup, the physician and nurses that were treating her realized, oh, wait, this isn't a stroke. This is an infection caused by a urinary tract infections, and she's experiencing the first symptoms of sepsis. And what we realized was this problem occurs quite frequently in the ER with postmenopausal women. And while digging into the problem further, we realized this is a really huge problem, occurs quite frequently, and there aren't really good solutions to treat this or to prevent this issue from happening. Women who experience UTIs are told to increase their water intake, to take cranberry extracts, probiotics, or apply topical estrogen creams, all of which are poorly tolerated by the patients and really don't solve the problem. So we had this need that we identified, a problem that we identified, which we knew hit a huge population and didn't really have good solutions. So we first tried to solve this problem from a device mindset, thinking, okay, maybe we could create a device that could sit locally in the bladder, deliver drugs and therapy, and treat the patient's UTI that way. And after really truly understanding the problem inside out from multiple perspectives of multiple stakeholders, so from the patient's perspectives, their caregivers, physicians, nurses, really diving into what products currently exist and what are the disadvantages of the existing products, and what are some of the underlying concerns of patients. We realized, through an iterative process, that device is really not the way to go with this. This is going to be a pharmaceutical product, a product that enables long-term extended drug delivery in the bladder and avoids the systemic effects of oral use of antibiotics. So that is what Watershed Therapeutics is working on. We're really focused on developing a drug delivery platform that could provide long-term relief from recurrent urinary tract infections while avoiding systemic drug effects.
Very laudable challenge, and I love that you came at it from an observational point of view. Was that how did you approach that portion of the discovery? Did you just spend days in the clinic, or was it a series of asking questions?
Well, so the observation was discovered first through just honestly doing like a 24-hour call in the ER and making these observations, taking notes of, patients' pain points. what they're experiencing, what the pain points are of the physicians and nurses that are treating them, what are their frustrations, and really where is the underlying problem. We also went into talking to other experts, urologists, urogynecologists, other folks that are experts in this area to understand really what are the disadvantages and advantages of current existing solutions here, and why hasn't anything come up that's better, to really understand how to go about solving this problem. And that really helped us develop a target product profile that could better serve not only the patients, but could also better serve the physicians as well.
Nice. Okay. Well, good. Well, you just mentioned target product profile. So let's move over back to Manjari. And I believe that's a pretty common term in the drug development space. So Manjari, as you spoke about the vaccine case earlier, and global health and some of the things that PATH is doing, but maybe can you focus in on some more details on what's going right, what kind of gives you hope that PATH and the work that you're doing is going to make an impact in a positive direction?
So maybe I can start by based on our collective experience enduring the COVID-19 pandemic. I think we've come to the realization that even though needles and syringes are inexpensive, however, vaccinating millions of people in the world is not an easy task. And so what PATH is trying to do is not specifically related to COVID-19, but in general, looking at alternate ways to deliver the vaccine. And with the goal being that everyone, again, coming back to equitable access, everyone gets the vaccine and leads a healthy life. So PATH has been looking at alternate, low-cost, alternate, simplified ways to deliver the vaccine. And one of the technologies which I'm very excited about and I would love to talk about is Microarray Patch technology. And I have a
Yeah, why don't you share?
Let me share that. Give me a second here. I'll let you know if you can see this. Are you able to see?
Yes.
Okay. So, so PATH has established a center of excellence for evaluating, identifying, and advancing microarray patch for administering vaccines and pharmaceuticals. So what you're seeing here on my screen is an example of a certain type of microarray patch. So microarray patch basically consists of very fine microscopic projections. They're applied to the body like a Band-Aid, and it's painless. And you remove the Band-Aid, and the vaccine is delivered. It is actually as simple as that. This specific microarray patch, which you are seeing here, the needles are dissolvable. So there's no there are no projections which stay after removal of after administration. So you apply, you remove, and the projections dissolve in the interstitial fluid upon coming in contact very rapidly. So this technology, this particular dissolvable microarray patch, I think you can see the schematic on the right, how it dissolves and you remove it.
So the needles there that are shown kind of a blue-gray.
Yes.
So are we just replacing a single needle for a bunch of needles? And is this going to be a painful experience for the user? How does that work?
So PATH is conducting technology feasibility, looking at currently we have several projects which are ongoing, focusing on evaluation of these patches. There's a technical feasibility component where we look at stability of the vaccines in this format. How do they perform? Do they maintain their antigen content potency over time? Then there is a human factors evaluation, which looks at exactly that. Is there a pain component associated upon administration? And so we are trying to quantify that by scoring the pain. We're also looking at whether it leaves any type of residue on the skin. Does it leave a mark on the skin, which may look like a grid pattern? And how long does that mark stay? And does it go away? Or does it stay like a bruise? So those are all the factors which are being looked into with each of these map technologies. So this is one example, dissolving microarray patch. There are other microarray patches which are, for example, they're coated with the drug or the vaccine. And of course, they perform differently. So you apply, the vaccine gets dissolved, you remove the patch and discard the way you would discard a syringe needle. So this is, but the technology, as you can see, very fine projections. So you don't feel the pain. This is the feedback which we have received. Don't feel the pain as much as a traditional syringe needle, but the verdict is still out there. This is the data which is being collected from the field.
And I imagine you could deliver a variety of medications this way. We were speaking about vaccine before. I assume, was this compatible with like a LNP mRNA vaccine formulation, or would that be a custom formulation that would have to be developed for this technology?
So again, each microarray patch developer is using their own drying process for coating the vaccine or depositing the vaccine. The process which they are using depends on the type of vaccine which is being coated. So for example, if it is a live attenuated viral vaccine, the process has to be gentle versus trying to deposit antiretroviral drugs, which are more tolerant to high temperature. So the coating process, deposition process may be different based on the type of vaccine candidate. Looking at the size of the projections, and I think you can also see from this, the bottom, the lower left panel, that the size of these microarray patches is very small. So if you are looking at loading small molecule drugs in milligram quantities, it will be challenging to have that loaded on a single microarray patch. You may need more than one patch, or you may need a larger microarray patch. So, it is not one size fits all. The maps may have to be customized a bit for small molecules versus vaccines.
Or much larger Band-Aids, I imagine. So
much larger Band-Aids.
Well, yeah, very exciting technology. Are there any things on the global health front that, so this is something that you're excited about? Are there areas where you've seen us stumble as we try to deliver healthcare to global health? I assume the maps are one way that you're trying to work around some of the challenges,
Yeah.
but.
Yeah, so this is one technology. Of course, the other considerations which go into selection and advancement of a technology, and that in general applies to all the technologies we look at, is the manufacturability, scalability, and it, as with any novel technology, that can be expensive. But what we need to look at is not so much as the cost per se of the technology. Look at the overall total systems cost. If you're getting a product which is thermostable, simple to administer, potentially does not require skilled health workers, the overall system cost will be lower. So, so it has to be taken, you have to take a holistic view when we are trying to introduce a novel technology in low and medium income countries. So that's, it requires input from, input on regulatory analysis, cost considerations, manufacturability, availability of material, etc. All those parameters go into the technology selection. So this is one technology Path is working on. The other technology is needle-free mucosal immunization. So instead of traditional syringe needle IM parenteral approach, what if vaccines could be delivered sublingually under the tongue as a drop or as a mini tablet? It will bypass the oral GI route of administration. Not only will you get a mucosal immune response, the first line of defense at the point of entry, but also a systemic immune response. So this is an area where Path has been working on for last few years. Very exciting. And so what you're seeing here on the screen is the Path developed a formulation for sublingual buccal administration. The formulation is freeze-dried. It's a freeze-dried formulation. So if you're familiar with freeze drying, traditionally freeze drying is carried out in glass vials or in bulk trays. Here, imagine freeze drying is carried out in a blister sheet where you can have the cavities of whatever size shape you want, and single step process, and you get freeze dried tablets. They do not require any further processing or coating of any sort, and the size is flexible depending on the target population. You just place it under the tongue for
Okay.
So this
Very
is
exciting.
Yeah.
You can see the potential scalability of this, obviously, this application.
Yes.
Okay, well, Deb, again, you had mentioned something about maybe talking about some additional delivery mechanisms as well, and I know behind the scenes that you've got some up your sleeve. So, what do you got? Show us. How else should we be looking at delivering these nucleic acid vaccines?
Yeah, we've always, if you think about what an ideal vaccine would be, and there's no such thing right now, okay, it's going to be one that's not just safe and effective, okay, they have to be safe, that's number one, they have to be effective, it's number two, or they're never going to get licensed. But on top of that, we want them to be stable at room temperature. And we want them to be ideally self-administered, at least be able to be administered without requiring, a specialized training to be able to do that. We want them to work in all demographics, including elderly and immune compromised people. We want them to be easy to distribute and to manufacture, very quickly to manufacture. So we're trying to work towards that ultimately. But one of the things I think that really needs to happen is in the realm of delivery. And let me just I'm going to just share my screen here very quickly, and hopefully get a little presentation mode here.
Yep.
I'm going to talk about one of the delivery technologies that I'm working on, and in disclosure that I'm a co-founder of a biotech company called Orlance that is working on developing this, and actually in collaboration with Product Creation Studios is helping us to do this. So anyway, the vision here for the basically is to achieve this sort of eventually needle-free delivery of room temperature stable RNA and DNA vaccines. I like to liken that to the Star Trek guy because the vision long-term is sort of very futuristic. The idea that you could just kind of point a device at somebody and say, hey, you're vaccinated, and so on. And over here really is a real device that Orlance, a biotech company I co-founded, is working on in collaboration with product creation studios. It came up with, helped us to come up with this particular design where, basically it's doesn't require any specialized training to administer the vaccine. You just push a trigger and that particular vaccine would then get administered into the skin. Now, I loved Manjari's talk about patches because I'm a huge fan of the skin, and I'll get into that a little bit. But this is how this particular technology works. And basically what we do is we formulate DNA or RNA onto gold particles. And this is actually an electron micrograph showing DNA on gold particles. It looks like a little gummy sort of thing, but that's actually how it precipitates on these microparticles. And these microparticles are 1 micron in size or less. Now, when you do formulate them, they're lyophilized, they're dry, and at room they are very stable at room temperature, whether they're DNA or RNA. And so that could support worldwide distribution. Now, then you take the device here that we just showed you here, and there's the way that actually we propel these particles is when you push this trigger, it results in releasing some helium gas that actually accelerates into the barrel of this gene gun here. And accelerates these particles at supersonic speed, and then they kind of get released out of the barrel there. So now you've got that needle-free delivery, and you have potential for self-administration or at a minimum to install one of these, at pharmacies or at various clinics around where, you don't necessarily need a particular training to administer it. It's just, you just need to push a trigger for that. And then when it gets actually into the epidermis, these particles get specifically delivered into the epidermis of the skin. And this is actually a histology showing these particles glowing here. They don't glow normally, but it's just the way the light is into the epidermis of the skin. And because these are so small, it is completely pain-free. Now, this has been in phase 1 and even early phase 2 clinical trials. And when we've asked the subjects to rank the pain from 1 to 5, or 5 is a needle stick, they always rank it at 1. It really doesn't hurt at all. Here, it feels like a puff of air. And so, there's a big issue with needles, in terms of getting compliance and getting people to take their vaccine. So if you have a pain-free vaccine, similar to the patch that Manjari talked about, or say deliver with the gene gun, you have a potential to improve compliance. So that's kind of how that works. And so through these particular technologies, we know that if it's DNA and RNA, they're very easy to scale up. It doesn't take much at all, except for, just some nucleic acids. We're working actually on, separate from this, a strategy where you don't even need, all you need, everything is scaled up in vitro. In a small tube, you can scale up 5,000 doses within 3 days pretty quickly, whether it's DNA or RNA. Now, when it's lyophilized like this and gold particles, it's stable at room temperature. For DNA, we know that it's stable for years. You just put it on the shelf and it doesn't, the biopotency doesn't decline at all. For RNA, we've just gotten started on that. And so we're starting to work on the stability. Obviously, RNA is less stable than DNA in general, but we're working on that. And then obviously self-administered, so we accomplish that. This is actually what the device used to look like before we started working with product creation studios. So it looks like something created in your garage, and that's because it was created in the garage originally. And so the idea, of course, with the nice, clean clinical device is to actually move this particular model into phase 1 human clinical trials by Orlance. And so just to kind of give you an idea then more specifically of how this works, I want to talk about the skin. The skin is really a unique site to actually immunize. And it's something why I'm excited about patches and I'm excited about the gene gun is because you think about the skin, it really is our first defense against pathogens. And unlike the muscle where most vaccines are deposited, it is chock full of antigen presenting cells. It is custom designed to stimulate immune responses. So if you put vaccines there, you can usually get away with a lot lower dose and get a lot better immunogenicity for them. The other thing that we discovered is that now our gene gun actually targets the epidermis. And of course, most vaccines are going to be delivered into the muscle here. But if you target vaccines into the epidermal layer, we found that actually you can stimulate both mucosal and systemic immune responses. So our vaccines, when they go in the epidermis, we get a nice systemic antibody and T cell response. But when we look in the lung, or if we look in the gut, we can actually find B and T cells localized there. We don't fully quite understand the mechanism, but we believe there's a crosstalk between the skin and our mucosal compartments that allows this sort of stimulation of both systemic as well as mucosal immunity. So this is actually just a schematic showing that once these particles go in the skin, this blue is just gene expression. That's the DNA vaccine expressing in the just in the epidermis of the skin. You can see that's where the particles are also delivered. Now, once they get in there, now as I mentioned, the epidermis is just chock full of these antigen presenting cells. They're called epidermal Langerhans cells. And you see the particles nicely associating with these Langerhans cells. So the particle gets inside the cell, and then the DNA or RNA elutes off of that particle and then gets into the, if it's DNA, it has to go in the nucleus. If it's RNA, it can sit in the cytoplasm. And then once it's inside the cell, that cell is going to translate that into protein and then present that to the immune system. And so then what you get is basically all of this antigens produced in the skin. Now that antigen presenting cell, it traffics to the draining lymph node where it then can interact with immune cells and stimulate both antibody as well as T cell responses, as we talked about. So that's really the nuts and bolts of how that technology works. But the idea ultimately is to try to address some of the gaps that nucleic acid vaccines have. And as we talked about the issue of requiring ultra code for the RNA vaccines. For DNA vaccines, the biggest issue were their poor immunogenicity. And one of the things that we found that when we actually use a gene gun to deliver DNA vaccines in the epidermis, that we can actually get much stronger immune responses than when you deliver the DNA into the muscle with 1,000-fold less DNA in terms of doses. And why is that? We think it's two things. Number one, we're getting direct intracellular delivery of that DNA into the cell, whereas other methods that just inject in the muscle results in that DNA going into the extracellular spaces, and then it's got to make its way into the cell and then into the nucleus. So we bypass that, getting right into the cell. And the other reason, as I mentioned, is that we're accessing a very immunocompetent organ. We're going into the skin as opposed to the muscle. And those two in combination, we think, make for a much more immunogenic DNA vaccine. And then, as I mentioned, because those gold particles are room temperature stable, we might have a potential of developing RNA vaccines that would be much more stable at warmer temperatures as well. So that's sort of the idea of where we want to take and sort of eventually get closer to coming up with a, what we would call the ideal vaccine.
And what are you going after? What diseases are really going to benefit from that additional mucosal response? And are you going after those diseases, or is it all COVID right now?
Yeah, absolutely. So as if you think about a lot of viruses, the way they access and the way they get infected is through our mucosal surfaces, either by sexual transmission or inhaled. And so influenza is a good example. We're developing a universal influenza vaccine. What we mean by that is one vaccine that's going to be able to stimulate immune responses against a lot of different variants of influenza. Similar to SARS-CoV-2, a big issue we have is the emergence of new variants. And so we're going to have to design vaccines that are going to be able to trigger immune responses that protect not just against whatever circulating at the time, but new variants that might come in the future or new potential pandemics. So that's the other feature which I didn't quite mention is that RNA and DNA vaccines are really exciting from the perspective that you can encode anything in them. If you want to make a multi-antigen vaccine that's going to stimulate responses against multiple different viruses, all you have to do is add an additional genetic code for those ones. And so they have that potential, that plasticity to be able to induce these broad responses against whole families of viruses and stuff. So influenza, SARS-CoV-2, actually combine SARS-CoV-2 and influenza vaccine that everybody gets seasonally once a year, rather than having to get flu and COVID separately. Other ones that we're working on are, for example, a therapeutic vaccine for HIV that's able to stimulate those T cell responses I told you about that can eliminate infected cells, because the problem with HIV is that it stays in our body long term. And so we need a way to trigger immune responses that are able to find those infected cells. and eliminate them. So so those are some of the examples, but there's a whole you name it, where you can probably make a DNA or RNA vaccine against it.
Nice. Okay, I see some questions are starting to come in on the chat, so keep those coming, folks. We'll get to yours shortly here. So, Saniya, turning our attention back to Watershed, we're kind of a nation of pill poppers, so why not just keep giving a person a pill a day and to manage this chronic condition of UTI? What's better about your solution?
Yeah, so I mean, UTIs are currently treated by use of oral antibiotics. And, the two major issues with oral antibiotics is the compliance part. You have to remember to take them every day and actually complete the entire course for it to be fully efficacious. The other part to it is the systemic effects that are caused by taking oral antibiotics. You're taking them orally. The drug has an effect on multiple organs. Women experience, issues with GI tracts and other sorts of effects that are not related to the UTI. And a very little amount of drug actually gets to the bladder where the therapy needs to actually go. So instead, what we have developed is a platform that can be easily and locally delivered into the bladder through the urethra. What you see here on the left is a prototype of, our drug formulation, and it consists of a mix of polymers with drug embedded in them, with an active ingredient embedded in them. And what we've learned through interviews with patients and interviews with physicians and actually seeing the problem at hand is that, anything that goes in the bladder will calcify. Anything that goes in the bladder could be felt because there's an area in the bladder region at the bottom of the bladder, which is called the trigon area, with a concentrated amount of nerves, that if anything sits on that bladder or sits on that area or causes pressure in that area, women will feel it, people will feel it. And it's actually nature's way of basically telling you that your bladder's full, you need to go pee. Whenever your bladder gets filled with urine, there will be pressure felt in that nerve area and you basically go void yourself. So the same way we realize anything that will sit in that area is going to be extremely uncomfortable. And there are a lot of products or products that have come out in the market which sit in that area. And the biggest complaint in patient-reported outcomes in clinical trials is that it's intolerable. And so what we've developed is a platform that floats to avoid essentially this area, the trigon nerve area, and slowly releases the drug in the bladder locally for an overextended period of time. We have prototypes that release drug for over the course of 3 months. And now in development, we're trying to extend that drug release up to 6 months. And as the drug is releasing, the polymer slowly degrades and disappears, and therefore doesn't require any sort of extra procedure to remove it. And so essentially the idea is that a patient would go in to a urologist's office, get this put in through a simple urethral catheter procedure, and basically you set it and forget it, and the patient is UTI free for over the course of several months. So there are no sort of systemic effects that are associated with oral antibiotics with this therapy. And at the same time, patients don't have to remember to constantly take their medication every single day. This is something that you set and it's doing its work in the bladder.
I love it. And it looks like you probably have some delivery challenges there, or has that part been figured out? Is it a simple syringe?
So right now we're using a Foley catheter, and it's a common procedure that's used to catheterize patients in the hospital when they're unable to urinate or you need to do a bladder washout. And these are just done through Foley catheters in various diameters. We can fit our product and deliver our product through a catheter as small as 12 French, which is about 4 millimeters in diameter.
Wow. Okay. Very impressive. Well, since we're talking to you already, again, a reminder, start putting your questions in, audience, so we can get to a few of those at the end. But for the last question, I always like to kind of get a glimpse of the future. So since we're already talking to you, Saniya, maybe tell us where is Watershed going? Where is this technology going? Is it a platform that goes beyond UTI? Will we be a healthier nation and world in 10 years because of your innovation?
I hope so. I hope that is true. We definitely are fully confident that that could be the case. UTI is not only a problem in the US, but it's a huge global epidemic, and antibiotic resistance is a huge problem as well, especially in these patients that are chronically using antibiotics to prevent their UTIs. So in that way, we're solving two really major issues. The really neat thing about our drug delivery platform is that we can actually combine any sort of drug in it. The process by which we're formulating this requires a thermal process. And so as long as a compound exists that can withstand high temperatures, so in this situation, or in our drug platform, a protein wouldn't work, or vaccine wouldn't work, but any sort of small molecule that has a high melting point could be incorporated. And so what we are looking to do is expand our drug delivery vehicle platform for treating other urological conditions like overactive bladder, interstitial cystitis, which is pain when urinating. But we could also apply this to other sort of clinical applications, issues in the GI tract, or other mucal adhesive conditions.
Beautiful, thanks. And as a mechanical engineer, I love it. There's just, hey, we figured out to make it buoyant, and that was kind of the key piece. I love it. So well done. Well, let's maybe Deb, we'll go back through and talk to you next. You know, you've kind of already hit us with Star Trek, so it's the future from yesteryear. What's the real future going to be like? Is it Star Trek? Where are we going with these nucleic acid vaccines?
Yeah, I think that there's really so much potential for both RNA and DNA vaccines. So one of the areas that that I think COVID-19 is going to help to open up. It's been an area of study for many years now, and we're going to start to see very soon. My prediction is the next RNA vaccine is going to be for influenza. It's going to be much better influenza vaccine. Right now, what happens with flu is that we try to predict what next year's flu virus is going to look like. And it takes right now current flu vaccines take about 6 to 9 months to produce once that particular next flu virus has been defined. But what happens is in 6 to 9 months, you could actually have the virus change again. And so then your current flu vaccines may not match up as well to the virus that is actually going around. And so that's why we have highly variable efficacy of our current flu vaccines, ranging from anywhere from 15% in 1 year to up to 70% another year, on average about 40 to 50% efficacy. And I think we can do much better with RNA vaccines, because you can actually, they're so fast, you can actually wait longer to actually define what is the composition going to be, because they only take, and we've shown this with COVID-19, 3 months from the time you've identified the virus to the time you actually have manufactured, scaled up and manufactured and have a vaccine ready to go into the population. And so that cutting that way back would mean that whatever vaccine is developed as an RNA vaccine is going to be a much closer match to whatever's circulating at the time. And so that's the immediate future. We're going to see that probably come up in the next year or two. The next thing that comes after that is what I call universal or pan vaccines. That's what I just talked about before. We're going to start to incorporate additional sequences within the RNA or DNA vaccine. And these sequences are going to be conserved among multiple different viruses in a family, so that you generate immunity, not just against whatever viruses happen to be circulating at the time, but you would have pre-existing immunity against other viruses that might pop up in the same family that could cause a future pandemic. And so that vision in the future for DNA RNA vaccines is they may prevent the next major pandemic, whether it's a coronavirus or influenza. And so the third area that I wanted to highlight, though, goes back to that whole second backup immune response that I mentioned, that let's say the antibodies are the viruses escaped the antibody response, and you've got this other arm of immunity called T cell responses, and these T cells survey infected cells and they're able to eliminate them from our body. Now, one of the things that I mentioned that we're working on in that aspect is for a therapeutic vaccine for HIV cure. Let's find those latently infected cells and eliminate them. But other things that we can think about is, for example, immunotherapy of chronic hepatitis B. That's another area that we're looking at. Yet let's take that a step further and think about cancer. There's a lot of similarities between the types of immune responses that are needed to eliminate a cancer cell and the types of immune responses needed to eliminate an infected cell. There's a lot of cross-fertilization in those two fields. But one of the cool things about RNA and DNA vaccines is that when you think about cancer, is there are a lot of times very much when we have a cancer cell, we're making these new sort of antigens that our body's not able to eliminate that cancerous cell, but it's tagged with what we call neoantigens, or basically cancer antigens. Now that can be very different for each individual. So sometimes you can't just make a cancer vaccine that's going to, you could for some aspects, but for the most part, one cancer vaccine won't work against all cancers or in all people. But what you can do is isolate the tumor from an individual and then actually identify what we call the neoantigens in the individual. So it's very specific antigens that your tumor is making. And then you encode that with the RNA or the DNA vaccine, and you inject that person with the RNA DNA vaccine. They start generating these T cells that can actually find and specifically eliminate the tumor cells. And that is sort of the next sort of frontier for DNA and RNA vaccines. It's ongoing right now. There are actually even phase 1 human trials starting in certain types of cancer, like for melanoma and breast cancer right now.
So using the body as the factory.
Exactly.
The perfect drug. Great. I love it. Manjari, last but not least, how will we change the face of global health over the next decade? You showed us a couple of ways, a couple of strong candidates. What will things look like in the coming years?
So I want to start by saying that RNA platform or RNA as a platform technology definitely has a lot of power and promise, and especially for infectious diseases. I mean, there's malaria, dengue, HIV. I don't want to say they are low-hanging fruit, but definitely more potential for infectious diseases. As therapeutics, I believe, again, that I think what immediately comes to mind is that using the platform for therapeutics perhaps need to identify the diseases which can be treated by causing expression of some protein. So maybe that perhaps is already ongoing. However, in terms of looking at it short-term, next steps versus long-term, I would again come back to, for short-term, I would say ensuring equitable access to vaccines is very important. And the way we do it, I would say a two-prong or a multi-prong approach. One, as we have noticed that in US, there's a lot of vaccine wastage, especially for the current COVID vaccines. And one of the reasons for that wastage is either poor stability, improper storage. And so once the vaccine vial is opened, it has to be used up immediately. And so therefore, so even though there is so much wastage here, unfortunately, those vaccines cannot be transported to other parts of the world because of this inherent issue with stability. So when I say multi-pronged approach, I think the immediate ask is, can we make these vaccines more second generation mRNA vaccines more thermotolerant? Can we do that? And because clearly the data has shown that these are highly efficacious and potent compared to any other type of vaccine candidate. So that is one. Then the other technologies or other ways to administer, I would say, is important to look at alternate ways to administer the vaccine, which is more simple. And I think what Deb presented, was fascinated by that, the gene gun approach. And I was trying to imagine, yes, having a centralized, especially for mass campaign, having that option, simple option to rapidly immunize large population, large sets of people. I think that is what is needed. So again, investing in new alternate ways to deliver vaccine is important. I know there are some groups working on nasal spray, nasal route for COVID vaccines. There are companies working on implants also. But again, highlighting that there is clearly a need to increase investment in alternate other technologies, which may be more effective.
Great, yeah. Thank you. And actually, we do have some questions in the chat here that are, maybe since we're talking to you, Manjari, Kelly's asking, are there innovations and techniques being created for low-income countries that could benefit high-income countries on cost and accessibility? So are you, because some of the things you described, the maps and the sublingual, seems like, boy, there's probably a lot of people in the developed world that would choose that path.
True. However, what we have seen is that in low and medium income countries, there is not much investment in R&D. R&D funding is very limited. So it is challenging for researchers in that part of the world to come up with innovations, although they are in a better position because they know the need. Unfortunately, that is not the case. Usually innovations happen here and then get transferred there, or at least the seed gets planted. A seed is planted here in the developed nations, and then the benefits are reaped in those parts of the world. So.
Right. Yeah, and then there's a question about RNA vaccines. Might they be freeze dried to maintain their efficacy? I think TBD, right? It sounds like the RNAs are delicate little.
I think Deb Deb can answer that better.
Yeah, that's fine. Deb.
Yeah, currently the lipid nanoparticles, they have to be in liquid. Okay, so they can be freeze dried. So when we think about actually making RNA vaccines stable at room temperature, we're going to have to get away from the lipid nanoparticle, and that's what we were thinking about. Formulate them lyophilized. They could be lyophilized and dried down, but then you have to have a way of getting them inside the cell. And so if you got rid of the lipid nanoparticle, you need some other strategy like the gene gun on particles. The issue with RNA is that it is quite unstable. And if you got RNases around, and there's RNases everywhere, they're right here in my room here. They're pretty much, we breathe them but they need liquid to be activated. It's an enzyme that has to be in an aqueous environment to be able to degrade the RNA. So if you get rid of the aqueous environment and you make RNA lyophilized and dried down, you could theoretically eliminate the issue of the RNases degrading the RNA itself. But they are a lot less stable than DNA, and that's where DNA might be a better solution for worldwide distribution. Now, India licensed the very first DNA vaccine. I think that they had this in mind when they were developing a DNA vaccine. unlike the RNA vaccine, it requires additional doses to get this similar immunogenicity, but that thing is stable. You can put that on the shelf, and I've taken DNA vaccines off a shelf 5 years later and still retain their biopotency, even they're stored at room temperature. So DNA vaccines may be the way to go for long-term stability, but we need to solve the problem of their immunogenicity. So, yeah.
And the problem with the immunogenicity is not enough, right? So
right. So the big difference between RNA and DNA, RNA is a natural trigger of innate immune. So innate immune responses have to get triggered to make an adaptive antibody response. And in RNA, our cells sense RNA as something that it needs to make immune responses again. So the minute RNA gets inside our cell, it starts to trigger this innate immune response, and that acts in a sense like an adjuvant itself to help the downstream activities happen. Now, DNA is not very immunogenic at all. It goes inside our cell and our body just kind of ignores it. So to solve that problem with DNA vaccines, we have to actually encode things within its sequence that would act as adjuvants to trigger those sorts of things. So that's one of the ways that we're trying to address the poor immunogenicity of DNA vaccines. Yeah. Got it. And since we've got you talking, what happens to the gold particles after being shot into the skin? Is it a permanent tattoo? Does it yeah, no, that's a great question. There's a little gold spot on your, so a lot of times when we've done the clinical trials, we just kind of shoot them like in the inner, arm, because you get a little brown spot there for a little bit. It disappears in 2 weeks. Basically, the gold particles are in the epidermis, and the epidermis undergoes turnover, so that most of the gold particles are lost, so they deliver their payload, the DNA. The DNA gets into the antigen presenting cell and starts to express that way, but the gold particles are lost with the shedding of the skin. Nice. And then we've got a couple of words of encouragement for Saniya. Brilliant job, Watershed. And then a couple part question down there. Interesting product, Watershed Therapeutics. Couple of questions. What happens to the drug when the bladder is empty? No urine to float on. Does it not gravitate towards the urethra and out then at that point?
Yeah, so good question. The formulation that we've made, we've been really careful about size selection. We wanted to basically develop something that could easily be delivered through a catheter, but that would be retained in the bladder long term. And the only way to do that is to put something in that's small enough to get in, but large enough so it doesn't come out. And our way of going about doing that was picking materials that are swallowable. So our formulation is made out of mix of materials that when they come into contact with urine will actually swell to 10 times their size. And so they do not get out of the bladder over the course of the duration. Then we have other embodiments where we've explored different shapes. So besides spheres, other shapes and embodiments, like, for example, cylinders or rods that will have the same sort of drug release profile, but because of their form and shape, retain better in the bladder. And in terms of bladder emptying out, if the bladder empties out, whether we have any sort of residual volume left or not, as the bladder fills up, the product will eventually become buoyant. So the rinse and void cycle of the bladder and the natural function of the bladder cycle doesn't really affect the product's retention. Nice. And Daria, if I mess up any part of this, please unmute and you can ask. But they're also asking, what about UTI with vesicular reflux, infection upstream of the bladder? Does the platform have any way to tackle that? So I guess upping the ante on the challenge here. Yeah, that's a really good question. We haven't taken this product into clinical trials just yet. We are scheduled to do that first in human trial before the end of this year. So we'll be able to better kind of understand if we can go after that sort of patient population that experiences UTI with vesicular reflux. But the way the platform works is it releases the drug and the drug dissolves homogenously in the volume of the urine in the bladder. And so if there is any sort of reflux, that reflux will be urine that has some amount of the compound or the active ingredient. And so thereby some sort of infection or some sort of bacteria could be exposed to that to that compound. We don't know that for certain, and again, has to be investigated further.
Nice. Okay. Well done there. And then we've got some additional questions about, Kelly's asking about the vaccine gun, and can we just do it with a bike pump to lower the costs? I think the helium is a key, as far as I understand, is a key part to getting the acceleration of the particles. But Deb, do you want to confirm that?
Yeah, no, you've hit on something that is a potential barrier, right? Because the helium is higher cost, and so that could be a potential barrier to getting that into lower income countries. So we've thought about that. And the helium, the reason why we use helium is because, you know, the function of the air is to accelerate those gold particles at supersonic speed, so they penetrate past the dead skin layer, dead pieces of your skin, and into the viable epidermis underneath. And to reach that accelerated supersonic speed, you have to have really light air, right? And so helium is one of those. You can't just use generic heavy air like from a bicycle pump. But some of the alternatives that could be considered, for example, hydrogen, that's also light air and much more cost effective and the like. So I think it's something down the line, once we've established this and, you know, gotten into, you know, clinical trials and, you know, as you know, worked out, you know, all those sorts of things that need to be worked out. Swapping out that gas, I think, is going to be one of the things that we'll have to consider. And I think it's very doable. It's just a matter of, you know, figuring out how to do that. Obviously, hydrogen has its own issues, such as flammability and stuff that would have to be addressed in terms of the safety part of things. But I think it's very doable and something that certainly we're thinking about in terms of what type of air. Mm-hmm.
Very nice. And Jenny's got a question about what are some of the current hurdles for mRNA vaccines for cancer treatment. And you spoke a little bit about that, Deb, but if anybody has comments on that.
Yeah, so one of the hurdles, just in general for mRNA vaccines for cancer, but also for infectious diseases, as we've all heard, because we're all now recommended come back for a fourth booster immunization, is that the immunity wanes fairly quickly, right? So, and we're constantly coming back for that booster immunization. For cancer treatment, ideally, we're going to want to have a little bit more sustained immunity to be able to eliminate those residual cancer cells. So one of the ways that our lab is actually addressing that is something called a Replicon Rna vaccine. So it differs from your conventional mRNA vaccine in that it's an RNA that also encodes an additional gene called a replicase. Okay, and once the RNA gets inside our cell, that replicase instructs the RNA to make additional copies of itself. So you get the RNA inside, and rather than one single strand producing X amount of protein, you've got one strand making 10 more strands of itself, producing 10 times more protein. And what we found is that actually allows the protein production to last a lot longer in vivo and actually prolongs that immune response. So that might be one way, that we can potentially address that hurdle.
Nice. Well, I want to take this moment then to thank our panelists, also to remind folks to hang out for a little bit if you've got some direct questions, maybe for the panelists, and if they can hang out for a little bit and be available, that would be great. We missed having this in person and kind of that natural networking and chatting that goes on afterwards. But we want to encourage that now. And but I do want to thank the panelists. Saniya, thank you. Manjari and Deb, this was very illuminating, and I know I learned a lot, and I thought I knew a few things about this stuff. So well done.
Thank you, Scott. Thank you for this opportunity. Yeah.
It was a pleasure. Pleasure talking.
Yeah.
I'm happy to take questions if there's any other ones. So.
Yeah.
Although I had a comment for Saniya there. I was thinking about your technology, use it for, one of the big things is inflammatory bowel disease and inflammation. And there's a lot of small molecule anti-inflammatories out there. Have you guys ever thought about formulating an anti-inflammatory alongside of maybe even the antibiotics? A lot of times with UTIs, there's an inflammation coupled with the infection as well.
We haven't looked in that space, but I'm glad that you brought it up. I'll definitely take a look.
Saniya, I also have a question for you. The picture which you showed, so in the vial, it contained granules, right? Is that the right polymer granules?
Sure.
Yeah, okay.
We call them particles, but you can call them
Particles.
granules as well.
Okay. And these are a few millimeters in diameter.
Yes, yes. They have been intentionally made to be smaller than a urethral catheter, so we can deliver it through a Foley catheter.
Okay.
They're anywhere from 2 to 4 millimeters in diameter.
Okay. And for when this is being administered, do you need, so what are you envisioning? Should there be a certain number of granules which have to be administered every single time to achieve the target dose?
So that's a great question. There's several factors that affect that, right? It's how much drug loading you can actually achieve in these particles and still maintain buoyancy. And really, you know, the trade-off here that we have to work with is the density of the drug, the density of the polymer, along with, you know, making sure that the overall formulation is less than the density of water, so that way they can remain floating. So that's definitely one aspect of it. The other aspect is bladder capacity. You know, we have a certain level of capacity we can hold. Normally, on average, we can hold anywhere from 300 to 500 milliliters of urine. And that really limits how much we can actually put in terms of product. Those are the limitations that we're working around. But the advantage here is, because this product is intended for local delivery, we don't need such a high dosage of drug that you would normally need if you're taking it orally. We can deliver a small amount that has the same therapeutic effect that you would try to get by taking a really large oral dosage. And the concentration that we're working in, or the therapeutic concentration levels that we're trying to work in, is parts per million or micrograms per milliliter. So it's a very, very small concentration to achieve a really impactful effect.
Right. Okay. Nice. Very nice. Thank you. Deb, I have a question for you as well. For the gene gun, these gold particles coated with DNA, how are these particles filled in the gene gun?
Yeah, so we actually have sort of what we call automated cart maker. So it's basically a very long tube that gets put in sort of what we call a spinner. It spins the tube. So we actually suspend the gold particles coated with DNA in basically ethanol once the DNA is precipitate on there. And then we pull up the slurry into a long tube, it's a tough cell tubing, and then we insert that actually into a machine that we've designed that actually is just automated. You just push a button, it automatically spins it at the right amount of time. And then once it spins it, so you've got all the particles on the walls of the interior of the tubing, then you just run some nitrogen through there to dry it out and elute off the ethanol from there. It dries for a little bit, and then you just cut them down to size that you need to actually insert into the device itself.
I see. So what is the how should I say, delivery efficiency, if I may use the right term? So are you, after each administration, have you looked at what is remaining? Do you get 100%, 90% delivery?
Yeah, it's between 90 and 100%, depending on the, I mean, things like if there's can impact the formulation, like if it's a lot of moisture in the air and stuff, that might make them retain a little bit more. So once the cartridges are cut up, they're actually stored in with desiccant to ensure that there's no moisture that collects in there. So they get loaded, I should say, on that reusable device. It's a 6-shooter, so just like a gun, and you actually load it in there. You can actually administer, up to 6 shots with the same. Okay. And you just really, what we do is we end up for the manufacturer, you actually preload the cylinder, and then you just pop that in the device, and you've got 6 shots ready to go. So.
I see. And the cylinders are then discarded, the empty?
Yep, those are disposable. Yep. Yep.
They're disposable. Okay.
Yeah.
Thank you.
Yeah. And I had a question for you too. I was wondering, how long do you have to leave the patch on the skin? Is that short or you have to leave it on?
I don't, I have the number, but I don't remember. It is not long. It is not double-digit minutes, but I don't know the exact number. I can get back to you on that.
That would depend on what you were applying to, right? If a vaccine versus I don't know, some other sort of drug.
No, I mean, if we are using the dissolvable needles, all we need is how much time it takes for the needles to dissolve.
Got it.
Yeah.
Got it.
Manjari, what's the biggest challenge for developing a patch like that?
So there is no single biggest challenge. I think scalability of the process, currently, that is one of the biggest challenges. How do you scale the process? And so if it has 500 projections, again, the QC part, ensuring that each projection is identical with identical loading, size, shape. So I think, yeah, so the manufacturing aspects are challenging, but I think the developers have, I think, have a pretty good handle on that. I think the question right now is, if you're talking about developing 100 million doses as needed for, mass immunization, how does that scale? So, so that is still TBD. I think we are waiting for the clinical data to come out. A lot of these are in phase 2 clinical trials with the vaccines, with vaccine candidates. Yeah.
Thank you.
All right. Dale, how are we doing on schedule? It looks like it's 5:30.
Yeah. So are we ready to wrap it up here?
Sounds like we could. Any last question from the team? We have panelists. You all have each other's emails now. It sounds like there could be some collaborations stemming from this, and would encourage that, of course.
With the three of these panelists, Scott, we don't need you as a moderator, right?
No,
this is
you
a
do not.
self-driving car we got here.
Excellent. That's the dream. The panelists ask each other questions, and I can sit there and learn.
All right, so let me do a little wrap up here, and we'll see if we can have a little time for networking. Well, let me first thank Scott and the panelists for joining us today. That was really great. Everyone stay tuned for in a few minutes. We'll have a little networking that'll go on. First, let's see. First, I want to remind everyone this event was created and produced by Life Science Washington, Come for the Savings, Stay for the Community, and in partnership with financial support from Product Creation Studio. Thank you, Scott and Product Creation Studio, for making this a free event today. And we will have another DeviceConnect later this summer. I think late summer will be our next one we'll shoot for. What else? We have Life Science Washington has several other upcoming events. Next week, we will have the World IP Day. And in June, we'll have an in-person event in Spokane, Washington for the East-West Summit. Go to the Life Science Washington homepage and click on events to find out more information and register. Okay, and now remember, this is the time for a little post-event networking. Our guests will be Scott and the panelists. Stay and say hi here. And what I'll do, I'm gonna turn off the recording here. And